Welcome to Summarize Science. Today, we're diving into a fascinating study about multiple sclerosis, or MS. Many people with MS experience cognitive issues, sometimes called brain fog, especially with processing information quickly. This study looks deep inside the brain, focusing on a critical relay station called the thalamus to understand why this happens. Researchers knew that MS involves two types of damage: distinct inflammatory lesions you can see on an MRI, and a more widespread, subtle degeneration. The big question was, how much does each type of damage contribute to the shrinking of the thalamus and the cognitive slowing that comes with it? To find out, they studied 100 people with MS, using advanced MRI techniques to measure the volume of 26 different regions within the thalamus, as well as the total amount of lesion damage. They also measured everyone's information processing speed with a standard cognitive test. Initially, they found that smaller volumes in 21 of the 26 thalamic regions were linked to slower processing speeds. But here's where it gets really interesting. The scientists then used a statistical model to essentially subtract the damage caused by the visible lesions, isolating the effects of the thalamus shrinking on its own. After this adjustment, 12 regions remained significantly linked to cognitive performance. As you can see in this image, these intrinsically vulnerable areas include sensory relay nuclei and important dissociative hubs. This chart shows the strength of that connection for each region, even after accounting for lesion damage. The ones with asterisks are the brain regions that are independently linked to cognitive function, regardless of the lesions. So, what about the other regions whose significance disappeared after the adjustment? This graph shows the answer very clearly. The brain regions that lost their link to cognition were much more heavily influenced by lesions, with over 34% of their atrophy being explained by lesion damage. In contrast, the intrinsically vulnerable group was far less affected by lesions, with only about 13% of their shrinkage related to them. This suggests two distinct pathways of damage are happening at the same time. Some parts of the thalamus shrink because they are connected to areas with lesions, a process called secondary degeneration. But other parts seem to be vulnerable to a primary, lesion-independent degeneration process, perhaps due to their high metabolic activity or specific cellular makeup. So, to recap the most important points from this study, first, cognitive decline in MS isn't just about the visible lesions. There's a hidden, independent degeneration process at play. Second, this process targets specific, intrinsically vulnerable nuclei within the thalamus, which are crucial for processing information. This helps explain why people with MS can still experience cognitive decline even when treatments are successfully stopping new lesion formation. What does this mean for the future? By identifying these specific vulnerable brain regions, doctors might one day be able to screen for patients at higher risk of cognitive problems. It also opens the door for developing new treatments that don't just target inflammation and lesions, but also protect these vital brain hubs from this intrinsic degeneration. Thanks for watching Summarize Science.